Добрый вечер всем, кто сегодня к нам присоединился. Сегодня у нас с лекцией выступит Евгений Викторович Кунин. Он расскажет нам об эволюции вирусов. Вы можете задавать свои вопросы в чате на YouTube, если вы нас слушаете, или в Библиобатон, если вы присоединились к виртуальной аудитории. Я буду ваши вопросы зачитывать. Евгений Викторович, вам слово.
Good evening, dear colleagues, ladies and gentlemen. It's a pleasure and an honor for me to talk to you tonight at the new faculty in my old alma mater. I'm very pleased to be there, so to speak, even if virtually. So we all live in the age of the coronavirus pandemic. We have been together in this ordeal for a year and a half. And everyone, at least everyone I know, and at least every biologist, I believe has become a virologist these days. But evidently, many people do not have experience as virologists, and so may not be fully oriented in what I like to call the world of viruses. And hence my lecture today. Which will try to briefly summarize the results of comparative analysis of virus genomes performed in my laboratory and others, and to give you an overview of the world of viruses and its global organization and evolution. I do understand that this is a lofty goal, this is an ambitious aspiration, but I think I want to try to convince you that in the last few years, we as a community managed to achieve considerable progress in understanding the overall organization and evolution of the world of viruses, to the extent that I dare say we now believe that this world, and it's indeed huge, as we shall see, is understandable to us. And we know the place of different viruses, in particular pathogenic ones, such as the latest coronavirus, in that world. So this is what we are going to talk about. So first of all, to begin with, an important message. It's by way of introduction. So viruses are really the preeminent, dominant entities in our Earth biosphere, physically and genetically. As in particular, and we shall talk about this in a little more detail soon, as in particular shown by virus metagenomics and widespread global studies of viromes from different habitats. I will use this shamelessly this word virome throughout my lecture, which means simply the entirety of viruses in a particular habitat, be it a human intestine or the entire biosphere. So the numbers are actually quite astonishing. Let us say 1 milliliter of seawater, depending on the particular habitat, its richness, contains from million to billion virus particles. And it's not only the numbers, not only the abundance, but the diversity of viruses that is striking. There are millions and millions and possibly billions of diverse species of viruses, in particular bacteriophages, in all kinds of habitats. Typically, not always, but typically, the number of virus particles in any sample from any environment exceeds the number of cells of any kind in the same environment by 1 or even 2 orders of magnitude, up to 100 virus particles per cell, making viruses indeed, at least by count, the most abundant biological entities on Earth. And the Earth-wide counts, accordingly, come to hyper-astronomical numbers, such as 10 to the power 31 virus particles present on Earth at any given moment, including the moment we speak. So to give you this superficial but curious comparison, there are about 10 to the power 24 stars in the observable region of our universe, and about 10 million fold more virus particles at any given moment on Earth. This is, however, not all that has to be said about virus diversity. In addition to this physical abundance and genetic diversity, viruses are remarkable also in that they employ effectively all imaginable strategies, all imaginable routes of transformation of genetic information. All cellular life forms, as of course everyone knows, follow the same route of genetic information transmission. From double-stranded DNA, which comprises the genome, to RNA. Evidently, some of RNAs are structured and others are messenger RNA, which give rise to protein through the translation process. The single route of information transmission. In contrast, viruses use all possible forms of nucleic acids as their genetic material, including RNA of both polarities, same as mRNA, and complementary plus and minus as they are designated, as well as double-stranded RNA, single-stranded RNA, and double-stranded RNA, and some very important viruses also use either RNA or DNA as their genome, but include a reverse transcription step in their replication cycle. Effectively, all possible variants, or logical and structurally possible variants of genetic information transmission are implemented by viruses, as summarized for the first time exactly 50 years ago by the great virologist Dna Baltimore, which suggests the idea that perhaps, just perhaps, virus-like entities descend directly from the primordial genetic pool and have been, so to speak, nature's original genetic test system for trying different ways to store and transmit genetic information. On top of this, since we want to understand the world of viruses and its evolution, we immediately must know two important things. One is that not only viruses are extremely abundant, they're ubiquitous. By that I mean that effectively every life form, every cellular organism is a host to more than one variety of a virus, with some very minor exceptions. The inevitability of viruses and other genetic parasites, in fact, can be almost proven mathematically, at least theoretically, let's say. We will not go there, but it's important message that genetic parasites are inevitable satellites of all cellular life. Another important issue to remember, though, is that viruses do not share a common ancestry. All cellular life forms share a common ancestry. Accordingly, we can build a single phylogenetic tree using, for that purpose, the sequences of universal genes such as ribosomal RNA or ribosomal proteins. There is no equivalent of these universal phylogenetic markers in viruses, which evidently makes the study of virus evolution a more interesting and challenging adventure on which we shall embark. I will also like to emphasize the very interesting distribution of viruses in nature by host range. In to show the distribution, we rely on these different so-called Baltimore classes, that is, viruses with different routes of information, different forms of genome encapsulated invariants and different routes of information transmission. And when we group viruses in that manner, we see interesting patterns that require explanation. For instance, in the virons of prokaryotes are heavily dominated, bacteria and archaea are heavily dominated by DNA viruses. In contrast, in the virons of animals and particularly plants, RNA viruses are even more common than DNA viruses. It's a challenge, an important one, I think, to understand the evolutionary cause, the biological and evolutionary causes of these differences in the global ecology of different types of viruses. So with that introduction, I would now like to go back in evolution nearly 4 billion years to the beginnings of life on Earth and discuss the concepts, scenarios, hypotheses, and I think the emerging understanding on the origin of viruses. So as a historical introduction, for many decades, in fact for about 100 years, I would say, origin and evolution of viruses has been discussed in the context of 3 types of evolutionary scenarios. To begin with the beginning, so to speak, the virus early hypothesis suggested that viruses indeed descend directly from primordial genetic elements, those that might have even preceded the emergence of cells, or at least of cells of the modern type. This diversity of the genetic routes, genetic strategies in viruses that we have discussed gives credence to these ideas. Now, the second type of explanation for virus origin are what may be called regression hypothesis, according to which viruses are the ultimate result of regressive evolution of parasitic cells. We all know that there exist parasitic bacteria, and moreover, some parasitic bacteria live inside of other cells, eukaryotic cells for sure, in some cases even other bacteria. So viruses, the obligate intracellular parasites, according to this line of thinking, are the ultimate product of degradation of such parasitic cellular life forms. Finally, the third group of explanations for virus origin and evolution, so-called escape gene hypothesis, according to which viruses descend from host genes, so to speak, normal genes of cellular organisms that somehow have acquired evolutionary autonomy and evolved into parasitic genetic elements and intracellular parasites. So the interesting question is, what can we say nowadays that we have so many genomes available? What can we say regarding the plausibility of each of these scenarios? Before I go directly into the discussion, I want to introduce our main tools, our main means for understanding and reconstructing evolution of the virus world. And these tools are what I call virus hallmark genes. That is, genes encoding proteins that play central roles in the replication and morphogenesis of viruses. As I noticed a few minutes ago, there is not a single universal gene that would be conserved in all viruses, which certainly complicates the study of virus evolution. But things are not too bleak because there are several genes, maybe about 15, 20 genes, that are quite widespread among different types of viruses. Hence the term virus hallmark genes. And some of these, like those illustrated on this slide, are shared between different Baltimore classes. For instance, RNA-dependent RNA polymerases, the key gene in the replication of RNA viruses, of which we shall talk in some details in a few minutes, reverse transcriptases, that is related and shared by all reverse transcribing viruses, reverse transcribing genetic elements, and different types of capsid proteins, like this so-called jelly roll capsid protein that is found in a great variety of viruses with icosahedral architecture of the capsid, and several others. The crucial point is that these are widespread, highly conserved genes in evolution of viruses shared by a wide range of viruses each. And by tracing the evolutionary fates of these genes, by building phylogenetic trees and exploring them in other details, we can have deep windows into the evolution of the virus world. And the deepest of those windows that we managed to open is into the virus replication modules. I say replication modules using this somewhat general term deliberately because this is not only about polymerases per se, but about other proteins involved in virus genome replication, such as endonucleases that are required for initiation of rolling circle DNA replication and some others. These are highly diverse enzymes which do not really show between different classes of viruses, do not really show significant similarity to each other. But when you do a more careful and deeper comparison at the structural level, we detect deep unifying simple unity, I would say, or unifying simplicity underlying this diversity of virus replication enzymes. They are all, strikingly, they are all these replication enzymes are based on the so-called RNA recognition motif fold, which is schematically shown here, a very simple, very basic protein fold that is extremely widespread in nature and found both in all these diverse enzymes and in non-enzymatic RNA binding proteins, such that the active center of enzymes is sort of grafted, if you will, on the RNA binding scaffold. So this profound unity of virus replication machineries implies that the origin of these goes back all the way to the primordial RNA world, where such proteins containing the RRM motif originally, RRM fold, originally might have been cofactors of ribozyme replicases, and only later, upon the transition from RNA world to RNA protein world, acquired the diverse catalytic activities. So, what we can see is that apparently viral replication machineries actually do go back to the primordial pool of replicators. when we look into the evolutionary provenance of virus structural proteins and morphogenetic modules, we see a very different picture. What we find here is that it is possible, again, primarily through comparisons of protein structure, because sequence comparisons are not quite adequate at this level of diversity. But by applying structural comparison, we can identify cellular ancestors for all major virus structural proteins. And these ancestors typically are carbohydrate or nucleic acid binding proteins, but different varieties with different or unrelated protein folds, demonstrating basically, I would say, that virus structural proteins evolved repeatedly and independently on multiple occasions in the course of evolution of life. Taken together, these observations provide for a synthetic, so to speak, chimeric scenario of virus origin, according to which the replication and structural modules of viruses have distinct origins. The replication modules originating from the primordial pool of different types of replicators, including RNA replicators, whereas the structural components have been scavenged from the cellular host at different multiple stages of evolution, some very early in the course of evolution, perhaps from some of the first cells, others considerably later. So in a sense, this is a hybrid of virus early scenario and the escaped gene scenario, which to the best of my understanding is by far our most credible view of the origin of viruses. So much for the ancient origins. Now I want to switch for a little while to more specific, even more practical matters, and speak about modern methodologies, about modern approaches that allow us to better understand the world of viruses, specifically about metagenomics, or when applied to viruses, we may call it metaviromics. So very remarkably, these days, the great majority of viruses that are being discovered in the biosphere in very, very different habitats, from the contents of our own intestine to hypersaline lakes and geothermal springs. Wherever new viruses are being discovered, the main approach, by far most important approach for discovering these viruses, is metagenomics, which means very simply, very simple to say, harder to do, but by now not even that hard to do. Simply sequencing all the DNA and then assembling all the DNA or all RNA from samples taken from different habitats. Orders of magnitude more new viruses now come from metagenomic studies than from the traditional laboratory cultivation approaches. Urging the International Committee on Virus Taxonomy to recognize officially that new viruses and new groups of viruses now can be recognized, can be formally recognized and established solely from sequence information obtained by metagenomic approaches. And by looking at this slide, you will easily understand why this is the case. why such a conservative body as the International Committee on Taxonomy of Viruses had no choice, even 4 years ago, than to adopt and embrace metagenomic virus discovery. Because when you look here, the horizontal axis on this plot shows the prevalence of different viruses and samples from the world ocean. That means samples were taken from a variety of places, stations, and the prevalence shows in how many of these stations a given virus was detected. And the vertical axis here shows on a logarithmic scale the abundance of a given virus genome. So on this plot, these colored little circles show viruses with known hosts. And the gray circles show viruses discovered solely by metagenomics. As you can see, both the most prevalent ones, number of most prevalent, and the most abundant ones, quite a few of these, and remember it's logarithmic scale, have been discovered by metagenomics only, have never been actually seen in any culture of any host grown in the laboratory. And this is a fairly old slide. It's about 4 years old. I'm just not diligent enough to update it. You would have seen an absolute preponderance of such gray circles if I bothered to update. So there's simply no choice other than recognize new virus taxa, new virus groups, discovered solely by metagenomics, because this is really the main path to the discovery of the diversity of viruses and prokaryotes, microbes for that matter, these days. So having established the impact of metagenomic, metaviromic approaches on the discovery of new viruses and exploration of the world of viruses, let us now go to greater detail and discuss the expansion of the world of RNA viruses, so the RNA domain, if you wish, of the virus world. So once again, metaviromics is decisive here. It reveals enormous diversity of RNA viruses and facilitates, or I would say enables, reconstruction of the global RNA virome evolution. So how do we reconstruct the evolution of the global RNA virome? By performing comparative and phylogenetic analysis of the only universal gene in RNA viruses, the one that encodes RNA-dependent RNA polymerase, RDRP for short, which is the key enzyme of RNA genome replication, which is homologous to reverse transcriptases as well. So this is we now deal with many thousands of these sequences, and this is not a very simple exercise because they are extremely highly diverse, despite their central role in virus replication. So we have to come up with a fairly sophisticated bioinformatics pipeline to derive this phylogenetic tree for an RNA-dependent RNA polymerase. This is again a couple of years old. Now these pipelines are even more complex, but they still work. And they produce phylogenetic trees. We shall look into greater details of the trees in a moment. Right now, I just want to show you this general overview that emphasizes that in this work that we published on 3 years ago, about half of the sequences from distinct virus groups came from metaviromics. Nowadays, it will be, I think, over 90%, as we shall discuss a little in a moment. Now, if you look at the specific structure of this tree, we shall see that, fortunately, despite the sequence diversity, there is well-definable structure in the tree. In particular, there are 5 stable major branches that in the new taxonomy, to which we will come in a few minutes, probably, I mean, not probably, they actually correspond to phyla of RNA viruses. 3 of these include primarily positive sense RNA viruses. 1 includes double-stranded RNA viruses, and the 5th one includes negative sense RNA viruses. Each of these major branches, 1, 2, 3, 4, 5, are quite stable and robust to the addition of new viruses. The relationship between them is less stable, less obvious, but at least there is defined structure in the tree, and evidently, when showing this tree nowadays, I cannot avoid relating to what is going on in the world, and so I map here some of the most important pathogenic viruses, including coronaviruses, hepatitis C virus, influenza, and Ebola, which belong to these different major branches in the tree. In particular, coronaviruses are lodged right here. They are distantly related to well-known but more picorna viruses such as polio, which are and caliciviruses, which are also important pathogens. Viruses with much smaller genomes. Interestingly and importantly, they are also related to plant viruses in this branch with even smaller genomes, indicating both the ancient origin of each of these major branches in the RNA virus evolutionary tree, and also most likely pointing to transfer of viruses between widely different hosts in the course of evolution of the virus world. We also can, at least tentatively, with a degree of confidence, reconstruct the scenario for the evolution of RNA virus genomes. And what we see here with some clarity is the trend for genome complexification in the course of evolution, starting with very simple precursors, ancestors typically encoding just the RNA-dependent RNA polymerase and the capsid protein, and proceeding to greater level of complexity by accumulating various kinds of protein domains, in particular acquiring from the host on 3 different occasions RNA helicases that enable replication of larger RNA genomes. Remarkably, the pinnacle of that complexification of RNA virus genomes are none other than the infamous coronaviruses that contain this large array of different domains, including those involved in proofreading and repair during RNA replication, which further facilitates the evolution of large genomes as well as separate genes for structural proteins and some involved in virus-host interaction. So this was the state of affairs about 3 years ago. Now, last year, with our colleagues, we published a paper that brought what I consider to be a very, very remarkable result. Why it's so remarkable is that the analysis of a single large metagenomic sample from a single location in the estuary of the Yangtze River in China resulted in doubling of the diversity of RNA viruses as judged by analysis of RNA-dependent RNA polymerases. So we first cluster the sequences to avoid over-representation of some widespread ones, and then count these clusters. And in the previous study, we had about 5,000, below 5,000 of these clusters of RNA-dependent RNA polymerases. And in this study, we already had more than 10,000. Let me emphasize again, this is a large sample, but a sample from a single location, showing that at this level of granularity, the diversity of RNA viruses is simply enormous. Entirely sure. Okay. But the other side of the same coin is that despite this huge diversity, most of the new viruses fall into the already established branches of the tree. There are surely lots of new interesting families of viruses. Maybe there are new orders, even classes, but all of them fall within one of these phyla, indicating that while the overall diversity is enormous and we may not be seeing or may not have been seeing a few months ago any signs of saturation at the higher level of granularity, at the level of virus phyla and classes, we may already have the principal structure of the diversity in place and only need to accommodate the new viruses within already existing major taxa. Oh, let me say another word here. I will not show slides, but just to mention, we are now with my colleagues. We are now about to complete a new and much larger scale metagenomic study of RNA virus diversity from thousands of different virions from different kinds of places. And what we see there is about a 5-fold increase in the diversity of RNA-dependent RNA polymerases, which is much less than the increase in the total amount of sequence, which actually suggests that we may be starting to see the saturation of the diversity, giving us some hope that within a few years, we actually may have a very good idea of what the diversity is. All right, time flies, and this is what I had a chance to tell you about RNA viruses. Let's now do a quick excursion through the rest of the virus world. To begin with the relatives of the RNA viruses, the reverse transcribing viruses in which the only universal gene is the one encoding the reverse transcriptases, which is a relative of the RNA-dependent RNA polymerase. Without going into much detail, in this tree, we also see 6 well-defined major branches, of which 4 include retrotranscribing viruses with RNA genomes, such as the infamous HIV, whose genomes schematic is shown here, and 2 distinct branches include viruses with DNA genomes that, however, replicate through an RNA intermediate, including this simple but dangerous hepatitis B viruses, which apparently evolved from RNA genome retrotranscribing viruses on 2 different independent occasions. Now we move from the RNA domain of the virosphere to the DNA domain, and first say a few words about single-stranded DNA viruses, which have almost all of them small circular genomes, which, in a sense, recapitulating the chimeric scenario for the origin of viruses in the first place, which I have discussed, also have a hybrid origin, with the replication apparatus involved in the so-called rolling circle replication mechanism, originating from a particular, from several groups of bacterial plasmids replicating through this scheme. Whereas the capsid proteins in the evolution of single-stranded DNA viruses, these so-called jelly roll capsid proteins, have been captured from different groups of RNA viruses. So the evolution of this class of viruses is a good illustration of these multiple convergent groups of virus evolution and the distinct evolutionary phase of the replication and structural modules. It also is to be noted that these single-stranded DNA viruses gave rise to these 2 families of small DNA virus, papillomaviridae, polyomaviridae, which also include important human pathogens, human and animal pathogens, including oncogenic viruses. And the last but the largest part of the virus world is the double-stranded DNA biosphere. These viruses are extremely diverse. They all employ the same route of information transmission, the same as the cellular host. But they show enormous diversity in terms of the size of the genomes, the genome and virus particles, the composition of the genomes, what they actually contain in their genomes, and their host range, and everything. These viruses actually do not share a single universal gene. I said before that there is not a single gene that would be universal in all viruses, but even in the double-stranded DNA part alone, there isn't a single universal gene. And so, in order to dissect that part of the virus world, we have to turn to different types of methods, in particular network methods that analyze networks of gene sharing and cluster genomes, in this case, genomes of viruses by shared genes. This works reasonably well, and we can detect several distinct modules in this network. 2 small ones, one including polyoma and papilloma viruses, evolved from single-stranded DNA viruses. We have already talked about them. And the others, this one, very mysterious, strange viruses of hyperthermophilic Archaea. That don't really have the time to discuss them in detail. They're very interesting, taking all kinds of beautiful shapes. But the two large modules that can be delineated in this network are these two that consist of two distinct unrelated large groups of DNA viruses. Which one? This one includes all the very well-known bacteriophages, tailed bacteriophages, or lambda, T4, etc., etc., along with animal herpes viruses, which are quite related to the bacteriophages if you look carefully. And the other one is highly, even more highly diverse, and includes, in particular, all the now famous giant viruses of eukaryotes, such as Mimi viruses. Sorry, a bit slow here. Okay, so it's time to make some conclusions from this necessarily brief presentation on the overall organization, overall structure of the world of viruses, which I believe are now starting to have a fairly good picture of. And this picture has been translated into taxonomy, or as we sometimes say, mega taxonomy, comprehensive taxonomy, that has been formally adopted by the International Committee for the Taxonomy of Viruses. This taxonomy at the highest level consists of these four taxa known as realms. And one of these is Riboviria, which includes RNA viruses and reverse transcribed viruses. Remember, they all have the same universal gene. Another realm, Monodnaviria, for single-strand DNA viruses and those small double-strand DNA viruses, and two distinct realms of double-strand DNA viruses. These are the four vast virus realms. And just very recently, a few months ago, we have formally introduced two new realms of viruses, much smaller, but very distinct and interesting, each of them. One, the so-called Rebosi viruses that employ ribozymes for certain stages in the replication of the genomes, including an important human pathogen, hepatitis delta virus, and the other known as adenoviruses, one of the largest known group of these unusual DNA viruses of hyperthermophilic Archaea that incorporate A-form DNA into the virus particles. If I may project into the future, I would suggest that we should expect the discovery of and formal recognition of quite a few additional small realms of viruses, but no major ones anymore. So at the lowest level of granular, at the coarsest grain, I think we already know the structure of the world of viruses. I will just list through the next few slides. We cannot go into the details, but what I want to illustrate simply is that we had the detailed taxonomy for each of these virus realms. And here we have the Riboviria, RNA viruses, reverse transcribing viruses, single-stranded DNA viruses, and the two realms of double-stranded RNA virus, double-stranded DNA viruses. So to give you a quick summary of the evolutionary relationship in the virus world as we now see them. What I do on this slide is superimpose the structure of realms of viruses based on evolutionary analysis and the Baltimore classes, which are based not on evolutionary analysis, but on a formal classification of pathways of genetic information storage and transmission. And what we see here is a fairly complex network held together, not surprisingly, by virus hallmark proteins, whereby several Baltimore classes, such as all those RNA birth transcribing viruses, comprise a single realm because they share key homologous gene, whereas other Baltimore classes, such as double-stranded DNA, are split between different unrelated realms. Again, certainly this picture will become richer, more complex with the discovery of new small realms, but the main contours, I think, are clear. So after presenting you this general picture, what I want to say quite emphatically is that we may know the principal structure of the virus world, but that does not make things boring. So many interesting and important things remain to be discovered. In particular, major new groups of viruses. And as an example, as a case in point, I want to account of the most abundant human-associated viruses. So the most abundant human-associated viruses are not any pathogens at all. These are, on the contrary, these are bacteriophages. They are not even viruses that replicate in our cells. They are bacteriophages that infect bacteria in our intestine. And most remarkably, these most abundant viruses in associated with our bodies remained completely unknown until recently, until a few years ago, when the metagenomic analysis have come of age. Because nowadays they have been grown in the laboratory, just a couple of these viruses. But not at the time, because they were just completely under the radar until they have been assembled from metagenomic sequences, and this highly abundant bacteriophage that in many humans can comprise up to 90% of the virus sequences came to light. On top of being completely unknown before, this virus genome seemed extremely unusual, with over 80% of the predicted proteins encoded in the approximately 100 kb genome, showing no significant similarity to anything in the database. So there was no opportunity to classify this virus. Fortunately, in a few years, the situation changed thanks to improved methods of genome comparison combined with growing databases. And the result of this analysis of these growing databases, we managed to discover an expensive family, or higher taxon probably, of bacteriophages that included at the time several hundred bacteriophage genomes. We were to different degrees, sometimes distantly, related to the crossphage, showed substantial diversity within themselves, but they have become less mysterious, and the structural modules have been identified, as well as the main genes involved in replication, and it became possible to build a phylogenetic tree of this expensive family of bacteriophages. in a very latest study, we actually greatly expanded even that diversity, now including thousands of bacteriophage genomes assembled from various metagenomes into this group. More importantly, discovering various novel ways of phage genome expression, including the usage of alternative genetic code and suppressor RNAs, the insertion of numerous introns and inteins in genes, possibly some completely novel ways of gene expression, to emphasize that the discovery of new viruses is not simply a chase for greater diversity. It allows one to discover new biology. This is what I wanted to tell you about the overall organization of the virus world, but you cannot stay blind to what is happening in the world and in society. So a considerable amount of my the effort in my lab in the last year and a half has been devoted to the study of the evolution of the novel coronavirus causing the COVID-19 pandemic, in particular adaptive evolution of the virus, and so a few words about that before I finish. Oh, something happened here. Something happened here. The way I am presenting my slides, I'm hoping you still can see them. So the key point here is that in order to understand the evolution of SARS coronavirus, we need to explore an unprecedented number of genomes. This work has been completed some 4 months ago, and at that time we analyzed about 300,000 complete genomes. Now it's the count goes into millions, complicating the analysis further, but also making it more informative. The things to emphasize here that we can see from these trees is that trees have structure. There are distinct major partitions whose evolutionary fates during the pandemic are different. Mostly what we detect in the evolution of these genes is purifying selection, indicating, not very surprisingly, that the virus is already well adopted. But we also do detect a small subset of sites strongly enriched in the spike and nucleocapsid protein genes that experience recurrent mutations apparently due to positive selection. So we can associate distinct mutational signatures with different partitions in the coronavirus tree. And the important thing that we can detect here is the small but robust, strongly supported network of co-occurring positively selected mutations, emphasizing the importance of epistatic interactions. What we also see very clearly is the region-specific differential evolutionary dynamics of these different partitions of the virus, emphasizing the different fitness and as well as their very distinctness, their distinct evolutionary phase. And the final thing that I want to, the final aspect that I want to emphasize here is the readily observable, steady diversification and possible speciation of the coronavirus, at least prior to vaccination. So it's very clear that during, it's not entirely trivial and very clear that during the pandemic, the diversity of the virus increased. The overall diversity is not like a single, at least until the introduction of vaccination, that a single isolate dominated the world. The diversity was increasing, suggesting the emergence of new variants that indeed has been observed. Hopefully, vaccination, massive vaccination breaks this trend. I think I will actually stop here after making quick conclusions. I try to show you that viruses are ubiquitous and enormously abundant in the biosphere, comprising a vast virus world. We have a great, still progressing, advancing way of investigating the world through metaviromics. We may be settling on a stable overall scenario of virus evolution and coarse-grained virus taxonomy. On smaller scale, however, highly abundant and important viruses remain to be discovered, as exemplified by the discovery of the expanding group of grass-like bacteriophages. And finally, this is all macroevolution, so to speak. Finally, it's important to note that microevolution analysis does inform us of the evolutionary regimes of pathogenic viruses and reveals virus adaptation, which is essential for understanding epidemics. And on my last slide, I simply acknowledge several colleagues from my evolutionary genomics group at the National Center for Biotechnology Information at NIH, Bethesda, and several key collaborators without whose work nothing would have been done. Thank you very much for your attention. I'm delighted to address any questions that you might have.
Thank you very much. We have some questions. The first question is it possible that different Baltimore types of viruses had different origins?
According to these hypotheses, I can read, yeah. Theoretically, this is surely possible. But fortunately, in our investigation of the origins of different viruses nowadays, we are not limited to speculation. We are doing and have done a lot of comparative genomic analysis. And all that comparative genomic analysis for each of these Baltimore classes points to the primordial origin of the replication module and later origin of the structural module from cellular ancestors. The specific origins are indeed different. Let's say the virion components originate from different cellular ancestors.
Thank you very much. And
my pleasure.
One more question in Russian. Как можно оценить число вирусов в клетке? Именно вирионов. ПЦР расскажет о последовательности вируса, но те же последовательности могут быть и в геноме ДНК эукариота.
Но, в принципе, конечно, могут. Значит, если речь идет об РНК, то обычно вычитается все, что присутствует в ДНК. Если речь идет о ДНК, то, конечно же, можно и в клетке, и не в клетке, кстати, в любых образцах из окружающей среды, где люди выделяют фракцию вирусных частиц или даже более точными методами там, центрифугирования в градиенте хлористого цезия выделяют вирионы, и потом можно подсчитывать.
Спасибо большое. Еще у нас есть вопросы.
Абсолютно.
Except from relatively conservative proteins, viruses have a lot of unique proteins. Do you have any suggestions? How can we guess their functions?
Well, indeed, viruses with larger genomes have a variety of genes that are either less strongly conserved only in small groups, or look orphans, that is not similar to anything. As for guessing the functions, well, highly sensitive sequence analysis and even more so structural analysis is very helpful in terms of at least understanding the general functional class, let us say. In particular, these days we have methods like AlphaFold and the latest editions of Rosetta, which have become very proficient in predicting protein structure without direct experiments. And such approaches are very helpful for at least understanding, which proteins are likely to be DNA binding, which proteins are likely to be structural proteins, which ones belong to what general functional class. Guessing specific functions is quite hard, especially when it comes to those virus proteins, and there are many, which are involved in what may be called counter defense, in inhibiting and activating host defense systems. We can try to predict in general that this is what these fast changing virus proteins do, but the specific functions require experiments.
Thank you very much. I would like to ask maybe a bit strange question. Are there any organisms or maybe species or higher groups of organisms that do not have viruses at all?
Oh, there are virtually no organisms that do not have viruses at all. The only, let's say as far as known to me, the only clear cases are intracellular parasites and symbionts, intracellular symbiotic and parasitic bacteria, which indeed often have not always by the way, but some of them, the more reduced ones, may have no associated viruses. Other than that, tell me, of course, if you know, but I'm unaware of any organisms from which, upon a more careful search, it wasn't possible to either isolate actual viruses or detect traces of virus genomes in the genome of the organism itself. So, to the very best of my understanding, viruses are indeed ubiquitous in nature.
Thanks a lot. One more question.
We have something else, some interesting longer question here.
Yes, and we also have questions in YouTube chat.
Yeah, please, as many as you like. No, but let me see this. Thanks so much. Yeah. How do you explain the observed pattern of replicomes, you probably mean, of different architectures throughout DNA virus? Why bacterial DNA viruses or replisomes code for both the Archaea eukaryotic, while Archaea and eukaryotes code almost exclusively for Archaea. Yeah, I think there's something a bit incomplete here, and they don't really, well, it is indeed a very interesting feature in DNA viruses is indeed the dominance of what may be called the Archaea eukaryotic replisome, and in particular DNA polymerase of the B family, as opposed to the bacterial type of replisome. Although this is not absolute, there are some viruses that encode POC, DNA polymerase 3, the main replication enzyme of bacteria, but indeed, the biosphere is dominated by what may be called Archaea eukaryotic type of replisome. Why? The shortest answer and simplest is I don't know. The more interesting one, perhaps, but also more speculative, is that perhaps, just perhaps, this type of replisome is just evolutionarily more ancient, coming regionally from the primordial pool of replicons to which viruses have been already adopted from a very early stage of evolution. Obvious speculation, but I don't have a better one. Can we learn anything about the origin of cellular replisomes from genomics of viruses? Given so much exchange between viruses and cells, especially when the replication components are involved, I'm afraid that learning about the origins is not origins of cellular replisomes is not entirely straightforward. Okay. Maybe should be asked to immunologists, but are there any insights why some viral infections causes very strong immunity, while others do not? For instance, both measles and SARS are quite high, but measles does cause very strong persistent immunity, unlike SARS. Yes, this is a complicated, difficult question. I don't think anyone in the world has a definitive answer. I think here devil really is in the details, and more specifically in the relative flexibility of virus capsid proteins, capsid and other structural proteins that elicit neutralizing antibodies. For instance, the hemagglutinin of influenza viruses is a particularly flexible protein structure that is therefore extremely apt at escaping immunity. So such relatively small structural details of virus protein may be very important for the immune reactions. So we have said that the origin of viruses might be inevitable. Could you elaborate on that more? What laws, govern the emergence of viruses? Are there any kinds of do this happen on different scales of the organization? These are good questions. I could point you to some literature. I wouldn't elaborate in every detail, but very briefly, in terms that indeed relate to evolution of complex systems in general and different levels of organization. Yes, I believe that parasites, the emergence of parasites is inevitable at different levels of organization, and we actually see them at different levels in nature. It's not just a guess. Why? Because effectively, in the state of the cellular state, the state of an autonomous cell is a comparatively low entropy state. They're put very, sticking to the statistical sort of Boltzmann understanding of entropy, there are few ways to make a functioning cell and many ways to destroy it. On the other hand, there are many different ways to simplify the system and create a biological entity that is dependent on more complex entities. So the parasitic state is a high entropy state compared to the cellular organization. And accordingly, parasite-free states of organisms are evolutionarily unstable. Parasites emerge indeed at different stages and different levels of organization. Such is the general theoretical framework. I may point you, if you want to, specific literature.
Thank you very much. I think we can switch to another question. Yeah?
We can.
Someone's typing. Okay.
Someone is typing, yes.
Is anything known about the role of bacteriophages on the hosts of their hosts? Do bacteriophages of human gut bacteria have an impact on human health?
Beyond doubt, they do. And the most obvious way they do that is by changing the composition of bacteria in the microbiome, killing off some hosts, or accordingly allowing the blooming of others, etc. Whether or not bacteriophages have a direct role, let's say being directly consumed by the intestine cells, is a more interesting and complicated question. I don't think we have a very good information on that, but it cannot be ruled out. But in any case, being major component of the microbiome, at least by affecting its composition, they definitely impact human health.
Thank you very much. Еще один вопрос по-русски. Евгений Викторович, планируете ли вы написать еще какую-нибудь книгу, а то логику случая мы уже прочитали?
Это любопытный вопрос. Ну, вы знаете, человек планирует, а жизнь располагает. Я-то планирую, но в процессе, так сказать, достаточно интенсивной работы, да еще когда наваливаются такие вещи, как пандемия, с которыми тоже приходится работать, к сожалению, эти планы не удается осуществить так быстро, как хотелось бы. Посмотрим. Планы есть, но посмотрим.
Хорошо, мы будем ждать. One more question. A question about SARS-CoV-2. Do we have any concepts about the freedom on the fitness landscape of spike protein? is the number of highly infected variant is principally limited?
That's a good question. There is no doubt that it is limited. The question is, what is the limit? Now, I can just brief you about a recent study in the lab that now only exists as a preprint, where we addressed a very related, directly, specifically addressed a related question. Namely, how likely is the emergence of breakthrough antibody escaping variants from different variants of SARS-CoV-2? In particular, the wild type Delta and Gamma variants have been compared. Evidently, in order to escape antibodies, the virus has to solve a trade-off problem. That is, retain efficient binding to the receptor on the low free energy binding to ACE2, while ceasing to efficiently bind neutralizing antibodies. So by protein modeling and calculating the interaction energies using the latest incarnation of the Rosetta suite, we tried to assess the possibilities for the emergence of such breakthrough mutants. And I should say that in this case, at least, they clearly see those limits. There are, in the aforementioned landscape, there is limited number of paths that each variant can take to reach that, so to speak, goal of escaping antibodies by retaining the high affinity for the receptor. And the different variants are not equal. They're located in different parts of the landscape, and they're not equal in terms of this potential. In particular, the potential of the Delta variant is less than that of other variants, including Gamma, which is, in a sense, maybe not surprising, because in general evolutionary terms, because when a specialist evolves that particularly well binds to the receptor, it's becoming progressively more difficult to escape binding of antibodies. So the number of paths on the landscape towards a more aggressive phenotype and phenotype with certain properties such as antibody escape is surely limited. And moreover, unfortunately, this potential decreases with increasing fitness. The higher you get on the landscape, to speak in oversimplified terms, the fewer opportunities you have. Have we already reached these? I mean, the virus reached these limits or not quite? We shall see.
Thanks a lot. And one more question. Are there any studies connecting the metaviromics and metagenomics data of cellular organisms? Is it possible
to but of course.
Which uncultured microbe hosts which virus with unknown host?
Yes, of course. Many researchers strive to establish these connections from, including ourselves, from metaviromic and metagenomic data. One really good way to identify virus-host associations is CRISPR. evidently only in prokaryotes, when you detect a CRISPR, several CRISPR spacers matching a given virus genome, you clinch the case. This is the host. Other methods are less direct, but for instance, when you detect highly similar genes in viruses and in certain cellular organisms, that, depending on the level of similarity, that also could be a strong indication. And then there are less direct methods for studying nucleotide composition and so on and so forth. But of course, there are many, and simply co-occurrence of certain viruses with certain hosts. There is a plethora of methods of different precision, and people actively work on this.
Thank you very much. I would like a question concerning viroids. What is the position of viroids in the evolutionary tree of life? Are they? Position of viroids. Interesting question. Viroids. I will elaborate in a moment. But typical viroids are unrelated to any virus. That is very clear. As you know, they're the smallest existing genetic parasites, pathogens. Some of them, they do not encode any proteins. Where do they originate from? So they are not linked to the virus, period. Where do they originate from? That's very unclear. There have been ideas on the origin of viroids from primordial RNA, primordial genetic elements. Personally, I think this is unlikely, given in particular the limited spread of viroids in nature, at least as far as we know. We are now actively working on discovering new viroids. They might originate from cell splicing, introns, or some other mobile elements like that. I think this is the most likely route. Now, very interesting critters are riboseviruses, such as hepatitis delta virus. These are circular viroid-like RNAs containing ribosomes, but they also encode proteins. The origin of the nucleic acid protein of the virus. The origin of the proteins is unclear, but still, this is remarkable, showing a path for a viroid to enter the biosphere. Okay, we have some additional questions here, interesting ones. Are there any, okay, step by step, are there any viruses that target organelles like mitochondria and plant plastids with their own translational apparatus? Would they be more similar to viruses? This is to viruses of bacteria or to the viruses of the eukaryotic host? Great question. Yes, there are. For example, so-called metaviruses, which is a widespread family of viruses replicating in mitochondria of some plants, fungi, and protists, unicellular eukaryotes. And lo and behold, these indeed very clearly are direct descendants of RNA bacteriophages, leviviruses that have lost the capsid protein gene. Quite remarkably, there are, after entering the eukaryotic domain of life through the mitochondria and dwelling in the mitochondria, some of these viruses have escaped into the cytoplasm, and then some of them have reacquired capsid proteins, showing that there are really intricate pathways into and out of the virus sphere. There are some other, say, double-stranded RNA viruses replicating in mitochondria of unicellular eukaryotes, whose origin is less clear, but which
also might originate from still unknown bacterial RNA viruses. To the very best of my understanding, there are no viruses have been discovered that would replicate in animal mitochondria, which may or may not have to do something with the reduction of the animal mitochondrial genome. Okay, next. Can we actually observe viruses that originated from escaped genes or primitive parasitic life forms recently? According to those hypotheses, is the process of new viruses emergence continuous? Interesting questions. Well, I would say yes, according to those hypotheses, the process of virus emergence is more or less continuous. That said, with regard to the reductive scenario, primitive parasitic life forms giving rise to viruses, I think we have zero evidence of this occurring ever. There is that not so many firm divides in biology, but I think this one holds. No one has observed the transition from a cell, however simple and primitive, to a virus. As for escape genes, this is more interesting. Certainly not continuous, certainly not every week, or even every year, but we have observed multiple occasions. Observed by genome analysis. It's not realistic to observe this in real time. Maybe in some very complicated laboratory experiments of which I am unaware. But by genome comparison, we can observe this. I mentioned in passing, unfortunately. So little time, the evolutionary scenarios for single-strand DNA viruses that clearly originated on several, up to 10, probably more. We don't know everything. I'm sure there are more occasions by the same route, from rolling circle replicating plasmids and capsid proteins of different RNA viruses. By different versions of escaped genes scenario, viruses have evolved on multiple occasions, although I would, in any rigorous sense, I would hesitate to call the process continuous. Could it be somehow estimated whether all eukaryotic viruses they're inherited from the last eukaryotic ancestor, or some of eukaryotic viruses were acquired from bacterial or archaeal hosts? Very good question. Now, I am not entirely even sure about last eukaryotic ancestor. We are actually working actively on trying to reconstruct the genome of that last ancestor, LICA as we call it fondly. It's not a very simple task. That said, most of the groups of eukaryotic viruses have evolved early on, even though there has been a lot of virus transfer between diverse groups of viruses too. Diverse groups of hosts too. For instance, one of the large groups, largest groups of double-stranded DNA viruses, the so-called viruses that have the so-called HK97 capsid protein, all these tailed bacteriophages of which were myriad or myriads, and herpes viruses. So there is this discontinuity. There are numerous bacteriophages found effectively in all bacteria and many archaea. And there is a single group of eukaryotic viruses that, to the best of our current knowledge, which is improving, only infect animals. We don't know for sure, and who knows what surprises might be awaiting us when, say, viruses of unicellular eukaryotes are investigated to a greater extent, but the current preferable scenario is that the ancestors of herpes viruses invaded the early animal lineage directly from bacteria. And I don't think this is the only case. So the answer is that some, yes, were acquired later from bacterial or less likely archaeal hosts. Okay, gene transfer agents, GTAs, have emerged on multiple occasions. Do you believe they are a specific adaptation for genetic recombination? Yes. They are specific adaptation for, yes, you may say recombination. I would be more specific. They are called gene transfer agents, not for nothing. This is what they do: transfer genes between related bacteria and likely archaea as well. This is one of the altruistic mechanisms that exist in nature. Under stress, if a bacterial genome, say Rhodococcus, contains GTAs, gene transfer agents, in its chromosome, they're induced and start forming mini virus particles, packaging random segments of the bacterial chromosome. And once many of those particles are formed, the cell dies, bursts, and the GTAs infect neighboring cells and transfer these multiple pieces of DNA, increasing the chance of survival. So yes, they are specific adaptation for dissemination of genetic material.
Thank you very much. We have new
questions. Yeah, my pleasure. Is it possible that sexual reproduction recombination evolved as part of an arms race against viruses to increase their genetic diversity? I do not think, this is a big subject, evidently. Origin of sexual reproduction, I wouldn't mix it with recombination because, you see, so, forgive me, promiscuous forms of sex, horizontal exchange of genes very much exists in the prokaryotic world. What we call sexual reproduction in the eukaryotic world is a different story. Indeed, origin has to be explained. I don't think there is a single factor here. There is different ones, but you are certainly right in that arms race and increasing the genetic diversity to resist viruses can be one of the factors.
Thanks a lot. And a new question from YouTube chat. What do you think about the RNA world at this moment? Are there new results concerning ribozymes and their role in evolution process?
Yeah, well, first of all, they think that maybe I should better say I, but I think many people in the community, in the part of the evolutionary biology community that is interested in the issue, think that in a sense, the RNA world is a logical inevitability. You cannot, this is the only route through which we can imagine the origin of life, circumventing at the first stage the requirement for protein. Now, the results on ribozymes. I should say this is not my main focus in any of my work, but yes, of course. There are new results. In particular, there are some very courageous researchers like Jerry Joyce in San Diego, who are trying all the time to improve ribozymes, to evolve new ribozymes in the laboratory, and certainly achieving some successes. The holy grail there, so to speak, is a highly efficient processive ribozyme RNA polymerase, and they are improving, but still, despite all the efforts so far, there is nothing even very close to being able to actually efficiently replicate other RNA. It's interesting that certain other important reactions and ancient reactions for sure, are much easier for ribozymes. Let us say amino acylation, formation of charged tRNA-like molecules. This is simple. There are many very small ribozymes that can condense with amino acids with good catalytic constants. So very different for different reactions. And of course, there is progress. What is the, we will never know how this happened in actuality. What was the actual role of this or that type of ribozymes in the origin of life. But evidently, the more can be done with them in the laboratory, the greater our confidence in the feasibility and reality of the RNA world. And as I say, even when a truly efficient, processive RNA polymerase made entirely of RNA will be made, that will be a great achievement, which will make us believe very strongly that there was an RNA some 4 billion years ago. Is it possible to reconstruct the morbulum of the proto-mitochondria? You mentioned metaviruses, for instance. Could we somehow test whether the metaviruses were acquired at the moment or these viruses were obtained later? This is a difficult question, and I would say it is quite hard to reconstruct the morbulum of the proto-mitochondria, because we really do not know. Of course, by looking at the virome of alpha proteobacteria, which are the ancestors of the mitochondria, we can have reasonable ideas of what it was. But the specific ancestor is not known. What viruses were specifically associated with it is hard to tell. That said, with regard to metaviruses, to me at least, it's very difficult and not sensical, frankly, to think about the possibility that these could have been acquired, in fact, these could have infected the mitochondria later as opposed to being inherited. There is no root, really. These viruses do not replicate in eukaryotic cells. So for them to penetrating the mitochondria, starting to replicate there at a later stage is a very tall order. So I think, we don't have a direct evidence of that inheritance, but I think the confidence runs high.
Thank you very much. I would like to ask you about the origin of virophages. Are they related to each other?
Well, yes. There are at least two groups of virophages that seem to have somewhat different origins. but they originate from a common pool of poorly characterized viruses of unicellular eukaryotes, which we dubbed Polinton-like viruses. They also gave rise to the so-called Polintons, which are virus-like transposable elements inserted into genomes of many eukaryotes. So apparently different groups of these palintoviruses have become virophages, have become parasites, have become parasites of giant viruses. But so again, generally, the virophages are, in a way, not a very special entity. Different viruses, on at least a couple of occasions, adapted to this secondary parasitic lifestyle. So if you pointed me to some literature on the emergence of viruses, you should quickly refer to, I think you, Dr. Chudovsky means the physical thermodynamic arguments for the inevitability of parasites. Yes, yes, I will point to that literature. Oh, I'll just send me an email and they will respond.
Someone is tapping now. But I think
the literature about CPR, candidate phyla radiation of bacteria, that is quite controversial. Is it, or does it comprise dozens of phyla? Oh, this is beyond the scope of today's presentation. To the best of my understanding, CPR is not a bad name, actually. It's a phylum, it's something like a super phylum that does have a common origin, most likely, but comprises, I don't know about dozens, but multiple phyla. Also, what can you say about mutualism in the world of viruses? Are there any such trends across any viral groups? Yes, there are. Well, mutualism. This is more complicated. Yes, you see, this is not sufficiently studied. There are several systems discovered in fungi, I believe in some unicellular eukaryotes that are indeed mutualistic. More common is commensalism or parasitism, whereby one virus is a satellite of another. There are many such examples among, let's say, plant RNA viruses, adeno-associated viruses, bacteriophages, others. There are many examples of those. True mutualism, I think there are just a few examples now, which does not mean that they don't exist. That must be investigated further.
коллеги, есть ли еще у кого-то вопросы? В том числе среди тех, кто слушает нас на YouTube. Похоже, вопросы закончились. Спасибо вам огромное, Евгений Викторович, что вы нашли на нас.
Вам большое спасибо за внимание. Удивительно приятно выступать перед такой заинтересованной аудиторией и отвечать на все эти замечательные вопросы. Огромное вам спасибо.
Спасибо вам огромное за лекцию и за вопросы. Спасибо всем, кто сегодня к нам присоединился. Запись нашей сегодняшней встречи будет доступна на YouTube. И вы сможете потом что-то пересмотреть, переслушать. Спасибо еще раз вам.
Да, ну и пожалуйста, кто без всяких сомнений и стеснений, мой имейл легко доступен. Продолжайте задавать вопросы.
Спасибо большое, всего доброго, до новых встреч.
Вам спасибо, всего наилучшего.